Hi, my name is Ahmad Fakri Abdullah. I'm from S1DT3.
Hi, my name is Nur Adila binti Muhammad Azhari and I'm from S1DT3.
Hi, my name is Alifa Elena binti Kasimain and I'm from S1DT3.
Translation is defined as a process of translation the genetic information in mRNA into sequence of amino acids in a polypeptide chain during protein synthesis. Translation is simple in principle but complex in its biochemistry and mechanics, especially in the eukaryotic cell. There is three processes involved in translation, which is initiation, elongation, and termination. Translation occurs in cytoplasm. For translation process, there are three molecular components involved in translation process that you should know, which is mRNA, tRNA, and rRNA. mRNA, which is messenger RNA, is a single-strand RNA that carries genetic information transcribed from DNA in nucleus of eukaryote. In cell of a prokaryote, mRNA is transcribed in the nucleus. Then, mRNA is transported out into the ribosome in the nucleus. Then, mRNA contains bases. This sequence of triplet bases called codon. Now, I will share with you about bases in mRNA. The bases in mRNA is guanine, uracil, adenine, and cytosine. It is denoted by the letters G, U, A, and C. Now, I will explain for you about tRNA, or we call it transfer RNA. Transfer RNA is produced in the nucleus of eukaryote. The 3D shape of tRNA are held by hydrogen bonds between complementary bases. The middle loop contains three bases are called anticodon, which are complementary to the codon for the amino acid it carries. In translation, tRNA at the ribosome join the amino acid to form a polypeptide.
In the activation stage, the correct amino acid is covalently bound to the correct transfer RNA tRNA. Amino acid is carried out by a family of related enzymes called amino acid tRNA synthesis. The active site of each type of amino acid tRNA synthesis is only a specific combination of amino acid and RNA. Regions of both the amino acid attachment and the anticodon end of the tRNA are instrumental in ensuring the specific fit. There are 20 different syntheses, one for each amino acid. Each synthesis is able to bind all the different tRNAs that code for its particular amino acid. The synthesis catalyzes the covalent attachment of the amino acid to its DNA in a process driven by the hydrolysis of ATP. The resulting amino acid tRNA, also called a charged tRNA, is released from the enzyme and is then available to deliver its amino acid to a growing polypeptide chain on a ribosome. The second instance of molecular regulation is the pairing of the tRNA anticodon with the appropriate mRNA codon. If one tRNA variety existed for each mRNA codon specifying an amino acid, there will be 61 tRNAs. In fact, there are only about 45, signifying that some tRNA must to be able to bind to more than one codon. Such versatility is possible because the rules for base pairing between the third nucleotide base of a codon and the corresponding base of a tRNA anticodon are released compared to those at other codon positions. For example, the nucleotide base U at the 5' end of a tRNA anticodon can pair with either A or G in the third position at the 3' end of an mRNA codon. The flexible base pairing at this codon position is called wobble. Wobble explains why the synonymous codons for a given amino acid most often differ in their third nucleotide base, but not in the other base. For example, of the tRNA anticodon, 3' use U 5' can base pair with either the mRNA codon 5' AGA 3' or 5' AGG 3', both of which code for arginine.
This example of the base pairing, the unusual nucleotide innocent can bind to cytosine, adenine, or uracil. This tRNA could then bind to 3 codons in the genetic code. There are numerous examples where all 3 would code for the same amino acid.
In initiation process, small ribosome subunit bind to mRNA and read along the mRNA until it reaches the start codon, which is AUG. Then, the initiation tRNA with anticodon 3' UAC 5' base pairs with the start codon on mRNA. The initiation tRNA carry amino acid methionine. After that, the large ribosome subunit then bind to the small subunit to form a complete RNA ribosome complex. The initiation tRNA is in the P site, while the empty A site is available for the next tRNA. In elongation process, there are 3 stages, which is codon recognition, peptide bond formation, and translocation. In the codon recognition stage, the ribosome moves along the mRNA in a 5' to 3' direction. New amino acid tRNA bind to A site in ribosome and is complementary to codon in mRNA. Then, hydrogen bonds are formed between mRNA codon and the anticodon on A site. The next stage is peptide bond formation. In this stage, the growing polypeptide chain peptidyl tRNA occupy P site. The amino acid from tRNA in the P site is removed and then become attached to the amino acid of the tRNA in the A site. Enzyme peptidyltransferase on the large ribosome subunit catalyze the formation of a peptide bond between amino acid in the P site to the amino acid in the A site. The last stage in the elongation process is translocation. In this stage, the ribosome moved 1 codon ahead. The tRNA was translocated in the A site to the P site. The uncharged tRNA, which is tRNA without amino acid, is moved from P site to the E site. From there, it exits the ribosome and joins the cytosol tRNA. The new mRNA codon to be translated is now in the A site, waiting for the next amino acid tRNA to bind. The process repeated until complete polypeptide chain is formed. Process, a stop codon in the mRNA reach the A site. The stop codons such as UAG, UAA, and UGA. Stop codon do not code for amino acid but it act as signals to stop the translation. Then a release factor come and bind directly to the stop codon in the A site. It hydrolyzes the bond between the tRNA in the P site and last amino acid of the polypeptide chain. All components of the translation process dissociate.
Now, I will share with you about polyribosome.
A single ribosome can make an average size polypeptide in less than a minute. However, multiple ribosomes translate an mRNA at the same time. That is, a single mRNA is used to make many copies of a polypeptide simultaneously. Once ribosome is far enough past the start codon, a second ribosome can attach the mRNA, eventually resulting in a number of ribosomes journeying along the mRNA. Polyribosomes are found in both bacteria and eukaryotic cells.
In conclusion, the process of translation is a part of the cell-central dogma system, helps in the tailoring an amino acid sequence from the gene. During translation, in the evitation step, the smaller and large subunit of ribosome bind to the mRNA transcript at its binding site. The process of constructing protein is started when the start codon AUG is recognized by the tRNA, that's UAC. During the elongation step, the phase of the translation, the mRNA triplet codon is read and complementary amino acid added by the tRNA. This is mRNA CAG is bind with tRNA GUC. The entire reaction is catalyzed by ribosomal RNA. Once the termination codon is reached, peptide tRNA stops the synthesis of the polypeptide chain. Interesting, I can say, in a process of translation of deciding the information present in the mRNA.